all the graduate students, I believe you probably have to get familiar about this because you want to get such as a constitutive form, hyperactive form, or get any transgene with a specific mutation. Then it will be important for you will be able to do it at the DNA residue. Then it will transcribe to become a new RNA to generate a new protein with a specific mutated residue you want to generate to put into your animal or plant cell. So that's today's basic principle I want to show you. So that's about two parts. Directed mutagenesis in the DNA level. Then I'm going to tell you how this kind of directed mutagenesis is to generate new protein. And even more modern biology, we will have other strategy to generate new protein, even not just mutagenesis. We will combine different modules to generate artificial component that never happen in evolution in real organism. And that probably can serve for industrial purpose. So that's today's major take-home message, and everyone should get familiar about this. And about the midterm exam, okay, midterm exam is very important to everyone, right? So I will give you some hint about my midterm exam will focus on which part of the lecture. I hope everyone will all understand and familiar about this part. And I will slow down to tell you, and this part is important, will be in your midterm exam. And you should memorize it and make sure you understand all the detail. Okay, I hope it's fair for everyone. Midterm exam will be short essay, this kind of format. So I basically will give you a few questions, such as what is cell-directed mutagenesis, and what is PCR-based mutagenesis. Some question like this. And then you need to write down a short essay for me. So is that clear? Okay. If not clear, you can leave a message in the panel or write me an email. I will answer you. Okay, let's follow up. So after the cell-directed mutagenesis, and then we will get a new version of protein. But in the past, most enzyme-based process for cell-directed mutagenesis is going to engineer the protein to fit specific purpose, such as for the detergent, you need to put some enzyme to make your detergent more powerful. But this enzyme still can survive in the detergent. So that will be a very difficult part, right? So they have a specific behind this kind of a genetic engineering for this technique. Okay, so synthetic biology or protein engineering, this field being awarded for the Nobel Prize in Chemistry in 2018 because three remarkable scientists' work contribute in this field. They basically contribute to the genetic change and the selection to develop a new technique for protein engineering that solve mankind of a chemical problem. So I will share their work with you guys later. Then you understand more about how synthetic biology and protein engineering contribute in biotechnology. Okay, so the first one, look at these three guys. Francis Arnold, George Smith, and Gregory Winter. Three of them are from different fields, from chemistry, from biology, and their work being awarded to Francis Arnold because of her idea about direct evolution for the enzyme. I will show you her work later. And phage display is a new technique for the screen, for the peptide and the antibody screen by George Smith and Gregory Winter. Three of these three guys' contribution in this new in this field to help us to improve all the protein engineering technique, to improve our technique to get a new protein to suit all, no matter in biomedical research purpose or industrial purpose. But today, I don't have too much time to talk about all the technique and new investment. I will focus on Francis Arnold's work, and then they will correlate with or more fit our today's lecture's purpose to help all the graduate students how to apply this kind of concept in your research. Okay, what's evolution? That's a very vague term, and almost it's still controversial, debatable in scientists, because during the evolution we think about selection pressure, about the mutation, so a lot of a different variety after reproductive system, and the reproductive development generate new oocyte, new sperm. After the fertilization, we will generate different variety of genes, different variety of proteins, but all those proteins may not pass the selection pressure. So some of them will be maintained, some of them will not be maintained in such a long history of the evolution. But you know, this kind of a process, it takes a long time, almost it takes forever to make a new protein to maintain in our population or in our society. But this kind of a robustness of the mutation is a major problem during the directed mutagenesis or for the protein engineering. We cannot hope a new protein, or we cannot wish a new generation of the protein through this kind of evolution to get one new protein, adapt our purpose or adapt our environment for this kind of event. So we need to have a more efficient way to get a new mutation, more efficient way to select suitable protein or select adaptable protein, adaptable gene to fit industry purpose. Why do I say so? Because those property or those fitness and those kind of a condition of a selection, it really rely on very long history in our society. So if you want to do something like this in the industry, almost impossible. So it takes forever to get the new protein right. So we cannot wait 10 years, 20 years, or even 100 years to get the improved property of the proteins. So why I say so? Because most of protein, they came from nature selection. If you think about it, it came from nature selection, they will have a native protein form. They will suit or purify from the real organism, from human, from pig, from cow, from monkey, or anything else, or from E. coli, from yeast. So all the native protein identify natural organism. You will see they will have a quite optimized survival physiology temperature to grow this kind of a protein, to grow this kind of organism. So those protein will only adapt from 18 degree to 65 degree. But you think about all the industrial purpose, all the company, this kind of protein could be useless because they cannot cannot be adapt a high temperature or even acidic condition. So the pH from most organisms will be nearly pH 7, but a lot of acidic solvent, right? Alkaline solvent, they cannot adapt this kind of a native protein. So most of the protein only can survive under aqueous solution, like a buffer system. So I believe all graduate students, when you start your first research work, your PI probably asks you to make the buffer. Why we need to make the buffer, PBS buffer, trace buffer, all kind of buffer, or for the cell culture to grow your egg chamber or to grow your cell, to grow your plant, because you would like to maintain the stability of those natural protein to make your experiment work. But you look at all the industrial enzyme of protein, you need to add into the animal feed. You need to add it from the dairy processing, fermentation, texture manufacture, or even to make a beer, to add into the detergent. So 50% of the enzyme will be applied into the detergent company to your laundry detergent. That's quite amazing, right? But most of the time we use a detergent to kill your cell, right? To purify your cell, to purify your protein, to denature the protein. How does it work? You still want the native, the enzyme can work with the detergent. That would be a big task, a big problem to solve. That will be the number one obstacle. So okay, so the Francis Arnold's work majorly started from here. They want to improve some lipase or protease enzyme to the fit into the laundry detergent. That's for stain removal. You can see that the Persil, you can go to the supermarket to read all the description about the Persil. They contain a lot of different lipase and protease to work together with the detergent and to remove all the blood stain, all the ink, everything, because they are very powerful to digest the protein. So it will be very powerful to digest all the blood, all the ink, all the oil contaminated on your clothes. So in the beginning, so Francis Arnold considered if we rely on evolution, rely on your luck, probably you will not get anything work. And then that detergent still need to adapt some kind of a DMSO, DMF, organic solvent, or detergent condition. And all those conditions will make your protein denature. So you think about lipase, protease. Most of them cannot be solved in the DMSO or DMF. So her team start to work on something like this. They start to work on a sublisting. It's a non-specific protease, but it will be very powerful protease purified from the bacillus. It's a sublisting, and then they hope go through mutagenesis to screen for any sublisting can survive in the buffer containing DMF. So after 4 rounds of the random mutagenesis, he got some mutated form of the sublisting, and then they will have a higher ability, higher enzyme activity compared with a wild type enzyme. So this kind of a successful experience, successful case, make her team become more, how to say that, more heroical way to try to improve all different kind of peptidase, protease, lipase to suitable to fit industrial purpose. So we are going to talk about her work started from here. And all those enzymes work together, become so powerful stain cutter, as I mentioned earlier, lipase or protease, because they can digest the protein, digest your phospholipid or any blood stain contaminated on your clothes. So it become very powerful one to work together with the detergent. Will be better than detergent, Triton or something only to do your laundry. And then, so all the protein engineering, they were started from the random mutagenesis or knowledge-based mutagenesis or computational protein design, and then through directive evolution, eventually to find out the suitable one. What does that mean? For the knowledgeable mutagenesis, it means you already know the potential protein residue that's required for the enzyme to digest this protein, or for enzyme to digest this lipid, like a lipase. You already know where is the catalytic triate and where is the covector binding site, and how protein to maintain the stability, where is the disulfide bond, right? So, but during this kind of a knowledge-based mutagenesis, basically you probably will not try to whole genome-wide mutagenesis. You will focus on specific residue to do mutation. But the most important thing, you will need the efficient functional assay to test wild type activity, mutation form activity, which one will digest your substrate better than wild type, or actually this mutation form will not do anything good to this new substrate. And the LAM, computational protein design, that'll be more modern design. We don't care about the protein residue. We just randomly do mutagenesis. Or even we make a new module to fit this substrate or fit this enzyme, such as we know this module, this domain of lipase is pretty powerful. Then we cut them down, make this new model. And then you want to target this model to another protein. And we probably will link this module with other protein-protein interaction domain, then together to fit into this new purpose. So computational protein design, we still based on our own knowledge about protein structure, protein domain, and the protein's function. But we probably will combine everything together, combine with a different protein together to make a new protein to assay its function. And so that'll be quite program specific. So you will do a lot of software bioinformatics prediction. Later on, then you will start to do this kind of experiment. Then direct evolution is like Francis Arnold. After the first successful experience, they start to have a more heroic way to do the mutagenesis. They don't care about the mutant or specific residue or interesting domain anymore. They just start to do random mutagenesis and random shuffling between different protein and eventually to make completely novel protein never exist in evolution or in animal kingdom or in plant to make a new function of the molecule for its purpose. That's a completely different concept. So I will show you a different case later. Okay, so but no matter knowledge-based mutagenesis, they have their own limitation, and direct evolution is also like this. But all of them will rely on a very efficient way to screen your end product. You need to come out very efficient way for screening to figure out this new protein is better form or the worst form. Otherwise, you will not get any successful result. Okay, so let's start from the so-called knowledge-based mutagenesis. So knowledge-based mutagenesis, the first part, the most difficult part, that's quite challenging about your knowledge. You have to choose which part of protein, which residue you want to change. Is there any good way to change? And after you make the choice, you have to figure out what kind of mutagenesis, what kind of technology you want to apply to efficiently generate new version of the protein. The third one, you want to evaluate these proteins variant to check whether they have improved property by screening or by selection. So, okay, let me make one thing clear. So everyone in this lecture room, anyone know what's a screening and what's a selection and what's the differences between these two techniques? One extra point for this answer. Anyone want to answer that? 什么事情叫 screening？ 什么事情叫 selection？ 还有什么差别或不同？ Okay, if you don't want to answer that, I will give you an answer, but you are going to lose your 1 point. After I finish my copy. Okay, screening. Basically, which means you have to check every one of your mutant form. Every mutant form, every variant, you have to check. That's called screening. Such as I'm talking about, I'm screening 2,000 undergraduate students applying my lab. So basically, I have to do 200 interview. So let's call screening. But selection, which means I set up a certain criteria. If they pass this criteria, you will get into the interview. Such as, I expect if your GPA is above 4, you will get a chance to interview with me. That will be selection, like an antibiotic selection. If they have antibiotics, amino acid resistant, they will survive. There are only a few colony, you will see that. If you want to check every variant, then to get the result, that's called screening. So that's a very different concept. Before I continue about my lecture later. So screening and selection, that's a quite important concept and to make this kind of strategy being successful. But how about the protein domain to change? You want to random pick up any residue to do mutagenesis, or you want to do some rational design, such as you already know where is the catalytic triad, so you probably just want to change that. Or you already know the disulfide bond, and you want to make it more stable, so add more disulfide bond. That's a rational design. But even no matter rational design or randomized mutagenesis, eventually, after mutagenesis, you still have to base on screening or selection result to see which domain or which mutant is the much better version for the protein. Okay, so this paper is quite simple. You can just take a quick look to see whole history about the protein engineering. So you can see how do they make the decision about which location they want to do mutagenesis, what kind of methods, such as what's the error-prone PCR, what's the saturation of mutagenesis, and what kind of a screening method they use. They use a substrate-based screening or high-throughput screening or any exhaustive screening or partial screening through the library. This different strategy will be very helpful for you to improve your own science project. And a different type of the mutation, such as a substitution, amino acid change, or deletion, insertion, or the most popular one, like a knockout homologous recombination, or different mutant form will give you different result or different structure for this protein. So all of them contribute for us for analysis of this protein's function. So usually for the genetics or biochemistry, we will analyze different form of the mutation to understand where is enzymatic catalytic site or protein-protein interaction domain so far. Okay, so you look at all the human history started from the protein engineering. The first thing we start to do site-directed mutagenesis from 1978. So this year is about 2024. So far, so many years ago, we already know that how to do it, how to do site-directed mutagenesis. Even 1988, the first monomeric insulin being designed, and then later on, protein folding pathway being described clearly. This part is very important because without this kind of knowledge, we don't know what is a suitable buffer and what kind of a domain folding structure, how to make everything work. So, and the 1992, we already can use hemoglobin as a blood substitute. So we already have some kind of artificial product for the temporal blood substitute for the emergency requirement. Okay, so then later on, the first chimeric antibody being approved for clinical use, even all the targeting treatment apply in the clinical purpose we use now is a chimeric antibody. I will show you guys a detail later. And the engineered insulin finally reached the market around 1996. So most insulin injection before the 1996, we get it from pig, and we have to take a risk from the contamination and the expensive in the all the charge everything. So So it becomes very difficult for the patient. So now we will have a recombinant protein to generate insulin, and even better, cheaper, and easy to manage. So that's a big milestone in this field. And then why choosing insulin start this kind of a no matter that a test or experiment has several reasons. First, insulin is quite small. Second, a lot of diabetes patients will need this product. So the pharmaceutical company would like to pay a lot of effort on this research. That's also a major reason. So recently, a lot of tailored enzyme, the so-called engineering enzyme, already start to be applied in our crop company, plant company. We always believe that we don't eat any GMO plant, right? But I have to say that's not real because we don't have a soybean. We don't plant a lot of soybean, but we drink soybean milk every morning in our culture, right? So a lot of this kind of soybean actually imported from USA, and USA already have a lot of GMO soybean on the market. So almost something that you cannot avoid. And so we have to think about all those kind of a problem advantage more carefully. Okay, so we are going to start from the method. That's also the major point for you guys if you want to pass this midterm exam. All those methods for the direct mutagenesis, for the side-directed mutagenesis will be the major point in your midterm exam, such as how we do the mutagenesis. We can do chemical mutagenesis by feed them EMS or feed them any kind of chemical to generate new mutation. But today I'm going to share with everyone is about error-prone PCR, side-directed mutagenesis, and the homologous recombination. These three methods, and then they'll be very popular in the lab or in the market to generate new mutant. So I believe it will be very useful for everyone after you finish today's lecture. Okay, computational strategy for high quality library design. That'll be more close to the bioinformatic process for the direct evolution. I will leave later to talk about this. Okay, let's start it from the very beginning. What did we do direct mutagenesis? We think about that, that's quite common for a graduate student my age. We have to start everything from the beginning. What do you mean? We have to actually grow M13 bacteriophage in my test tube. Why we use M13? That's a filamentous bacteriophage. It's a circular they can generate circular single-stranded RNA. When they generate single-stranded RNA, I can fit my original template with a new primer. And that mismatch new primer is the site I want to do mutagenesis. So after this primer, a neo with the original template with only one mismatch site. Because of E. coli, because if you feed the clinal fragment as a DNA polymerase, because if you feed the DNTP, they will fill out all those gaps to make a single-stranded DNA. Then you ligate those DNA with T4 DNA ligase, transform back to the E. coli, then ask them to make more plasmid for you. And eventually, theoretically, you will get a two single-stranded DNA. One is a parental strand, original M13, original template. And then the other one will be new point mutated template you design, right? 50%, 50%. But the problem is the 50% recovery rate for the design mutation never happened. Actually, we get less than 1% of the fetch. This kind of a M13 bacterial fetch to carry mutated gene. Okay, anyone would like to ask answer this puzzle? Why we only get one person? Anyone? 有没有人想知道为什么？为什么你就只有一 person 不到1分哦？如果被当掉的话，快要被当掉，这1分是你需要的。Pair with a wrong mismatch. Very close, but what do you mean? A wrong mismatch. What would happen? Because of only one base pair is wrongly matched. Then, this kind of wrongly matched residue, why it cannot survive? Because only one person, because as a parental strand, right? As a daughter strand. Theoretically, 50-50. Oh yeah, 周家伟, congratulations, you got this and 曹廷伟, okay, 3 of them. Write me email. I will give you 1 extra point for the extra bonus. That's correct. Usually DNA, they are not that stupid, right? We do the replication all the time. So we will have a mistake. All the mistake will be recognized by repair system, even one mismatch. This kind of differences. But how do they know which residue is wrong? Because parental strand usually have a methylation, right? So they can determine, oh, this is a parental strand. That should be correct for our repair system. And that's a daughter strand. So because of this, they will base on parental strand and fix this residue. So one person will be something wrong in our repair system. They did not fix it. So that's only one person. Okay, so the problem is here. Parental strand could be a big problem, right? So is there any way we can remove parental strand? Or is there any way we can try to do anything to fix it? So during this kind of directed mutagenesis, we apply to enzyme. Okay, the same 13 bacteriophage, we will transform this M13 phage, that's your gene you want to change, to the DUT-UNG strand of the E. coli. What's the DUT? DUT mutant and the UNG mutant, they are defective form of the UTPS. And another one will be uracil and glycosylase mutant. I will apply these 2 mutant here. I will apply these 2 mutated form of the E. coli here. Because you see that DUTP, that's a key enzyme for UTP become TTP. So that'll be critical step for the nucleotide biosynthesis. Without this enzyme, all the biosynthesis, they don't have, all the DNA replication, they don't have a DTTP. So that's a big disaster, right? So they only can incorporate DUTP. That'll be terribly wrong for the DNA replication. Okay, when this kind of a terribly wrong, the UTP being incorporated into the DNA replication, try to avoid this kind of problem. They still have a UNG, this kind of enzyme, right, to cut it out for this kind of misincorporation to ensure all the DNA replication is correct. So according to the template, GATC pairing, they will need a correct residue composition and the quantum to incorporate correct residue. If anything wrong, they will be cut it off by UNG, this enzyme. So if the UTPS is defective and the uracil and glycosylase, this enzyme, both of them are defective, which means when the original template incorporate UTP, the repair system cannot do anything to correct it. So we can do something like this. When they transform into the DUT-UNG mutated E. coli, then we'll make the parental strand to incorporate a lot of U here, right? Any T will be misincorporated, UTP here. So we'll make your parental strand contain uracil. Then the second part, you want to add the oligo. You want to change for the protein residue to pair to anneal with your target gene. And to do the synthesis, you do a PCR, clonal fragment, and the DNTP. Again, you make a new strand here. So you will have a two strand. One parental strand already have a UTP on the top. And then new strand will be pretty normal strand, only one mismatch. And the same strategy, you use it T4 DNA ligase, clonal fragment, everything together after you finish the DNA polymerization and ligation, then transform into the, pay attention, it's a wild type E. coli. So the wild type E. coli can recognize this one is not correct because they are DUT-UNG normal, right? So they definitely can recognize, oh, this guy is not correct. So we have to remove it. So eventually, only your target protein with a mismatch can survive. So this kind of DUTP cannot be incorporated into DNA, so will be removed. So the wild type E. coli contain the enzyme to remove it. So they will remove all the wrong parental strand. Only new strand will survive. So that's one strategy. That'll be very important. Still used in the lab. Even sometimes you feel that's a quite old strategy. But if one day you have to make your own mutation, let's already have a kit. And sometimes they even have a methylation strand, specific enzyme like a DPN to kill all the parental strand. So the major point for this kind of mutagenesis, you can make a new mutant easily with a one mismatch oligo. You can do everything by PCR, by clonal fragment to fill out the gap. But the major problem here you have to solve is to kill the parental strand. So there are a lot of different strategies to kill parental strand. That's one of the strategies. I want you guys to pay attention. That's quite important. Okay. So another will be even trickier, but that's quite easy. Like your target gene here, right? So usually you will put on two antibiotic selection marker. One is ampicillin sensitive, one is tetracycline resistant. You just take advantage of two antibiotics. You can do something, anything by yourself for your own. Okay, that's a target gene. You want to do mutagenesis. That's one mismatch here, right? And you can change the ampicillin sensitive gene to become resistant by one point mutation. Also, the other way will change tetracycline resistant gene to become tetracycline sensitive. Just generate one mutation. So after the mutagenesis with the 3 oligo, its plasmid transforms back to different antibody selection marker, such as you look at this. You have the original parental strand. You will grow your plasmid in the wild type of E. coli with tetracycline, but they cannot resist ampicillin, right? Because they don't have an ampicillin resistant gene. They will show ampicillin sensitive. So ampicillin will kill this plasmid, but tetracycline can survive. But after the mutagenesis, your plasmid have changed into tetracycline sensitive, ampicillin resistant. So basically, your daughter strand, after the replication, only new mutated form can survive under ampicillin resistant. So this kind of ampicillin resistant, ampicillin sensitive will be one point mutation will change resistant gene to a non-functional ampicillin resistant gene, right? So you can switch back and forth no matter what kind of strategy you want to use. It doesn't have to be ampicillin or tetracycline. You also can use other antibiotics, maybe neomycin or anything else, to do the similar experiment. All the major principle would be kill the parental strain and to leave the new mutated target gene. That will be the major reason to make this kind of a strategy to become successful to get your protein. And then this kind of strategy, usually you can get 90% of transform E. coli grow on the ampicillin plate with a specific mutation. Okay. And then later on, all the directed mutagenesis rely on PCR. So that's a more current strategy. We use it in the lab now. So even I want to generate any point mutation, that's easy. We just synthesize one mismatch on the primer, right? And then do the inverse PCR. Usually we do the PCR from the left side, right side, and then they move, they go face to face to emphasize, to synthesize in two direction, right? But inverse PCR in two primer, it goes inverse direction to finish all the amplification. And then this kind of a mutagenesis, you can change this one residue and then to get the whole full length of a DNA fragment for your own. Okay, even you want to generate deletion mutant, you just leave a gap here. And eventually they will synthesize a DNA fragment with gap to generate mutation you want. Or you want to generate insertion mutation. All the insertion part, you can make it in the primer, right? Make it in the primer. So then you can do this kind of inverse PCR to get your insertion mutant. Or you want to make a small insertion, you can make it in your primer, right? The same strategy, you do the inverse PCR. So all the different kind of strategy based on PCR, you can think it by yourself, but all of them will depends on how this PCR will work in your hand, in your test tube. A non-directed mutagenesis is a more simplified way. If I don't know what kind of a residue I want to change during primer biosynthesis, I don't know which residue I want to change. I probably will put in this kind of order, such as usually G, A, T, C must be equal quantity during primer biosynthesis. But if I don't know whether this residue should be changed to T or C or any residue, I will put 94% of G here, but 2% of each of them, like A, C, T, each of them will have a 2%. And then I will get 2% of the GAG. Every G will have a 2% A, 2% of C. This kind of synthesized mixture of a degenerate primer. And I definitely need an efficient way for screening new enzymes activity to get the correct product, to know this residue to change to A will be better, or this one change to C will be better. If a change to C or change to A will be better, their enzyme efficiency will be higher. Am I right? So this kind of a degenerate primer also being applied when we cross species to clone sequence homolog, such as one transcription factor, they have a homeodomain. Usually will have a conserved protein residue, about 60 amino acid. This 60 amino acid will be about around 180 to 200 nucleotide base. But we don't know mammalian, this homeodomain protein, and the fly, this homeodomain protein, or grouper fish, or Arabidopsis, do they have the same homeodomain? But why they belong to the same protein family? Because they have a conserved domain. By definition, conserved domain means protein amino acid is pretty conserved. from 70% to even sometimes 90% of the residue are the same, are identical. But the third base of the Ubo sequence could be different. So we can use this kind of degenerate primer randomly put into any residue, any nucleotide to see this kind of degenerate primer, which one, when they do annealing with a different species, the primer will find out their own target with a high affinity. Then they will amplify the homologue for you, right? Okay, here, like some kind of target gene, you use all the degenerate primer. They may not be completely identical, but through this kind of mutagenesis, you will pick up your clone, and with similar homology. After sequencing, you will know that's your homologue or not. Or even we can use this kind of degenerate primer to generate different version of the protein with only one residue different, or two residue different, or any three residue different, or any G, they have a different variety. So degenerate primer is another strategy for directed mutagenesis. So it will give you from one mutation to several mutations in this kind of a direct mutagenesis. Okay. Another one will be randomized mutagenesis. We have no idea about which part to make a mutation. So basically, it will be pretty random. Then I'll be another strategy I'm going to introduce it to you will be error-prone PCR and DNA shuffling. Okay, let me ask everyone. Do you prefer I finish all the lecture without any break? Or you guys want 10 minutes break? No break. Or break. 10 minutes break. Okay, 10 minutes break. Come back around 3:10. Okay. Okay, we are going to start our second session soon. Okay, Parker, one of the students asked about, I have a question about why do some protein from the same family have a different function? I also would like to know, but I will tell you my idea about this. Is it related to conserved domain? Okay, so for the research, such as when we always would like to use a bioinformatics tool to say if there's a conserved domain, theoretically they should have a conserved function. We hope that this kind of conserved function will help us to understand this protein's function. We don't have to do any experiment anymore. Unfortunately, even transcription factor with the DNA binding domain, transcriptional activation domain, for the most of time, we still have to do experiment. Why? They always give us surprise, such as in my lab, we have some kind of transcription factor, but turn out it did not bind DNA at all. Even they look like they have a one conserved DNA binding domain. But they actually use this DNA binding domain to do protein-protein interaction for some other domain. So for some other transcription factor, and then let the novel transcription factor actually bind to DNA, recognize a specific sequence for that. But sometimes after we finish all the research, we probably will figure out, oh, because in this species, there are a few residue are mutated. So this DNA binding domain, it will not recognize DNA anymore. But then they use this new version of the transcription factor, new version of the DNA binding domain for protein-protein interaction. So that's the reason why we still have to do it one by one to test. Even the bioinformatics, those tools are very powerful, very useful, has a lot of advantage. Always is the first step. I start my own research to see whether it has a specific domain, specific function. We can guess or hypothesize its a function in vivo, but eventually we still have to go back based on our knowledge to design experiment to do. I hope I can answer your question, but unfortunately, I guess no specific question or answer for this will be really satisfied. Okay, the second section we are going to start about the randomized mutagenesis. When I say randomized, mean no bias. We can do it any place, any part for the mutagenesis. Depends on your purpose, but you will need a very efficient screening or selection criteria to pick up the right target, to pick up the right protein for you. So the first one will be error-prone PCR. So everyone have ever done PCR, right? No matter in your undergraduate lab practice or undergraduate general biology, biochemistry, or even now in your lab, you probably need to make your own PCR. So all the PCR, all the senior probably will ask you to mix up your template, your primer with a commercial buffer for this, in case you are going to generate any wrong residue in your PCR, right? But the error-prone PCR, the concept is completely opposite. We just want to generate wrong residue in your protein. So how am I going to generate wrong residue? So your senior OPI usually will ask you to do the mutagenesis with proofreading tag DNA polymerase, right? Why not use original version of the tag DNA polymerase? Why they ask about proofreading DNA polymerase? Because they don't want you to make any mistake in your PCR product. But the thing about error-prone PCR, they basically want to randomly generate mutation or wrong residue in your PCR product. So you want to use some tag DNA polymerase without proofreading activity. So that's lesson number one. If you want to generate mutation, but you still use those so overpriced, expensive tag DNA polymerase, that'll be wrong. So wrong, completely wrong. So use a cheap tag DNA polymerase will be good enough. Okay, second part. Usually, I would use the commercial buffer. You just add your template and your primer and the tag will be enough. So we want to make sure the commercial buffer with the correct formula of the buffer. They usually will give you a suitable concentration of the magnesium chloride and the equal amount of the DNTP. If you make a DNTP by yourself, if you don't pipette well, the molar ratio is not quite correct between GATC or something like this, just like a degenerate primer, right? I just show you if the percentage is not quite correct, it will cause a misincorporation of the tech-DNA polymerase to the template. And why the high concentration of the magnesium chloride will cause a problem? That's a basic component for the PCR. I bet that most of the graduate students never pay attention about this. That's how much of the DNA template we need to have it, probably from several nanogram to 1 microgram. You won't make anything more because you will want to have a specific amplifier. If you add too much template, all your primer will have a wrong annealing to the template because too much, they can anneal to any spot. So only very few template will be enough for amplify specific target. And then your oligo primer, and it will be around 10 to 20 micromolar. And the DNTP will be under specific concentration, about 2 millimolar. And the thermostable DNA polymerase, which means CAC, it depends on commercial enzyme activity, probably from 0.5 unit to 2 unit. And the magnesium chloride, that's quite important, especially buffer system, KCl, trees, NaCl, that are under physiology condition to suit tech-DNA polymerase activity. But the magnesium chloride will be quite important and cannot be replaced because, okay, usually will be 1 to 5 millimolar. If above this concentration for the DNTP, I mean too much of the DNTP, too much of the primer will cause problem. Why it will cause problem? You look at this structure. Let's say your template. Okay, let's say your primer. It basically will anneal, and then with the template, when tech-DNA polymerase bring template and the DNTP together to this pocket, and the magnesium chloride will be a cofactor to stabilize this pocket to help your primer to anneal with your template. So if you add too much magnesium chloride, so you will make this complex too easy to fit into this pocket, to bind too strongly, which means even the primer anneal to the wrong side is fine. They still can incorporate. Especially when you add too much of the DNTP above 250 micromolar, maybe 5 millimolar or something like this, some kind of very exaggerated condition, they will have too many of the DNTP. So the wrong site, wrong incorporation will be added into this pocket, such as theoretically if this place a G should pair with a C, but if you add too much of the DNTP, even U or A or T will be incorporated. And too much of magnesium chloride also can stabilize this complex. It will cause a huge problem. So all the optimized condition, concentration, or the PCR reaction follow up the same rule. But okay, let's go back to C. I don't want to make this PCR reaction so precisely because I want to generate mutation. I want to incorporate wrong case, wrong residue onto this new synthesized fragment. So do it in opposite way. Use tech-DNA polymerase without proofreading. Even they found, oh, that's a mistake. Doesn't matter. I still can read through. I use a high concentration, sometimes even higher than 8 millimolar, to make this complex so stable. I add unequal amount of the NTP to induce a misincorporation. Then I can generate this kind of a randomized mutagenesis product, a so-called error-prone PCR. Okay, that is super important because even now, no matter in company, in the lab, if I want to do random mutagenesis, that's still the number one strategy I will choose to use to generate new mutation, new protein. So that is super important. Remember that. Okay, so usually 1 to 2 millimole magnesium chloride, right? For optimize the tech. So increase it. Okay, that will be very helpful. Okay. The second strategy about DNA shuffling. The Parker just asked about why the same family protein, they don't function similar or even function very differently. So that's a big question in biology. But a lot of same family gene can help us to generate new protein. I give an example like a homeodomain here, okay? As the original gene, we can do random mutagenesis to have a library. Each one has its own point mutation. From mouse, from human, from fly, from C. elegans. Okay, then we start to do random DNA digestion. We'll have a lot of small fragments, and then because I only use one enzyme, such as a Hindi3, all Hindi3 fragment, it's like a playing car, and then you can shuffling all DNA fragment together to mix everything together. So this is DNA shuffling, then ligate with the DNA ligase, T4 DNA ligase, and then put it back to the E. coli to make a more template for us. Then I will have a originally as a one same family protein, but with a different mutation, and then shuffling together to generate new protein. This protein, even they contain original DNA template, like we always know the homeodomain from human, like isepsin, iris, PAX658 gene, this kind of gene. But they already have a very different domain after this kind of DNA shuffling. They'll become brand new gene. Even you go back to think about all the proteases, you can do the same thing. They all have a specific catalytic domain, but I don't know which one will be better. I will shuffling together. They probably will give us 3 cutting sites, 3 different domains. After this kind of a DNA shuffling, will generate 3 protein, a brand new version for that. So the homologous gene arm also can do something like this. After the DNA shuffling, you will get a multiple related gene fused together. Then you can do the PCR to generate a new form for that. So that's a DNA shuffling idea for the randomized mutagenesis. So basically, you cannot predict initially what kind of a protein after DNA shuffling or after the error prompt PCR, what kind of protein you will get. Initially, you cannot really predict what kind of protein you will get. You only have to rely on sequencing to know the final outcome you will get. But before you do any sequencing, you want to make sure this one is better or this one is better, right? So you need a very efficient way for screening or for selection. Okay, so DNA shuffling and all of them still rely on related gene or single gene. So you also can create this kind of a library for yourself, such as you want to create a DNA shuffling library for the DNA binding domain, or create a library for the catalytic domain of the protease, or create a library with different mutation, different point mutation for the lipase, and then for desired activity. That's our very important concept. You need to know how to assay your desired activity to generate new protein. Then we call it as a molecular breeding. Breeding, this term is from very old genetics. We want to make a more efficient way to make the milk from the milk cow. So we have to breed from different country, different species to make a very complicated cross, to make all this kind of breeding together. But the molecular breeding is different. We just use different DNA fragment because we already know this protein can do something good for us, for our purpose. Then we can do this kind of a shuffling, random mutagenesis to make a new protein for the desired activity. So this is called molecular breeding. So molecular breeding highly rely on randomized mutagenesis, highly rely on error-prone PCR and DNA shuffling. Okay, I already said this 3 times, right? Okay, so see that, let's go back to see all the mutagenesis. They all have their own advantage and a disadvantage. As a chemical mutagenesis, usually we'll use any kind of chemical to torture your animal or plant or E. coli or something, EMS, UV, and any bisulfite that usually will randomly generate mutation on the backbone of the DNA. But the problem is the mutation rate is very low because all the animal will have their own repair system. So, and all the chemical, they will have a very hot spot. So it will not be really randomized mutagenesis. Will have uneven mutagenesis spectrum, such as EMS always will prefer to generate certain type of a mutant, like a point mutation will generate stop codon or something like this. So it also rely on the dose, how much EMS you feed into your animal or E. coli or in your experiment. So that will not be very easy to control. Especially sometimes I really do not like this way because I'm also a normal human being. When you contact with this kind of chemical, you should be very careful. Any wound on your finger, on your hand, and go through this kind of chemical exposure will generate mutation on you. So for the researcher contact with this kind, handle this kind of chemical mutagenesis, should be very careful. Should be well trained. Otherwise, I won't suggest you to do anything like this. Okay, and some of the mutation strand, like E. coli or something, because of their repair system has some problems. So they also can generate mutation for you. But this kind of E. coli is not very easy to grow. So the mutation rate is also very low. Error-prone PCR is our favorite strategy. So it will permit a high mutation rate, and you can do everything in your test tube. So after random mutagenesis, you basically generate a library with a mixture of the component of your PCR product, then transform back to the E. coli, let E. coli to do everything for you. And site-directed mutagenesis, remember that's a very focused. You already know where is the functional group, relevant residue you want to do mutagenesis, and that will increase all the specificity. But sometimes when you think another catalytic triad should be changed, but life will find its way out. You feel that's a DNA binding domain that should be important, but unfortunately, they doesn't use this domain to bind DNA or other residue. After you change, it will give you better result. So most of the time, it may not be always the best strategy for site-directed mutagenesis, although we know those enzymes, those proteins very well. Okay, computational strategy. Basically, you rely on our knowledge to the protein conformation. How do they fold? How do they make the enzyme protein contact? And to make the new domain in computer program. And later on, you still have to do all the experiment in the test tube in the animal to confirm that's a good version of the protein. Homologous recombination is like a DNA shuffling or non-homologous protein that just mix up everything together. That all depends on a brand new protein without any specific purpose, without specific reason. You just want to rely on this strategy for the molecular breeding to generate new version of protein for that. So I really suggest everyone to take a look about this Nature Genetics paper. Let's summarize. Let's summarize all the major strategy, no matter advantage, disadvantage, and all the what should I say, all the chemical methodology or all the take-home message for different strategy during the direct evolution and the metagenesis. It's a very useful strategy for everyone. Okay, let's go back to the protein engineering. We'll talk about native protein industrial enzyme. As I mentioned earlier, all the native protein actually came from the natural organism, no matter from E. coli to the human being, from fish or from pig. So they only survive around 18 degree to 65 degree. Below or above this temperature, usually the enzyme activity will not be good. Or above this pH or below this pH, the enzymatic activity will be reduced. And they only survive under the aqueous solution because most of those organisms maintain in the buffer, right? Our cytoplasm basically is a big buffer system. They need a buffer for all these enzymes function to maintain its physiology. But I already said it several times in this lecture, all those detergent company, animal feed company, even beverage company to make a beer, to make a beverage, to make an animal feed, won't fit this kind of a condition. Okay, let's talk about the industrial enzyme market. According to the 2011 calculation, only in USA market is about 4 billion because I make the new enzyme for this. It occupy 4 billion of the market. 2019 is about 7 billion. It increased a lot because we use by the 2025, it will be 10 billion market. So that's the reason why you feel that your laundry detergent is not that important and very cheap in the supermarket, right? But a lot of detergent being used for different purpose in the hospital, in the industry, in the other laundry company. They really use a lot. So they will use this enzyme a lot. They will require a lot of different form of the enzyme. So let me give you, okay, this example will not be important in your midterm. I just give you an example why those enzyme, new version of the enzyme is important. You just think about that. You want a fancy denim jeans to wear it every day. Those denim jeans in the beginning, that's so expensive. Probably charge you $2,000 to $4,000 to give you a beautiful stone wash denim jeans. Why? Why you need so much money for that? Because it will need amylase, pectinase, peroxidase, acid cellulase to make this gene desizing, de-dyeing, bleaching, and eventually make it a little bit like a ore denim jeans. So it doesn't make sense, right? The original denim jeans is purely blue, but that's so dumb. No one will wear this kind of denim jeans on the street, especially for teenager. We want a little bit old, like old jeans from your mother's closet. So the companies think about all different way. Use this kind of a bleaching, stone wash, this kind of a technique. Actually, they use a lot of enzyme to remove excess dye, to make it more plastic, more elastic, more fit into your body. So it will really need a lot of enzyme to work on one simple denim jeans. So that's another reason people will use or company will use a lot of enzyme in this kind of a manufacturer company. And then this kind of enzyme definitely will not use in the normal condition, normal buffer, normal optimized pH. Okay, you look at all the protease using detergent, alcohol production, amylase is a beer making, cellulase used for alcohol or glucose production. All of them will be very important in fermentation or all the fabric company to make a new product. Okay, so you think about that. The protein stability will be the big issue, big problem. A lot of protein when they add into the detergent or add them into some solvent to work together for your purpose. it will cause the protein denature. That's the first problem they met. So we already know that the disaccharide bond will increase the protein stability. And then if we already know the protein structure look like this, can we add more disaccharide bond to increase the stability? If that's a midterm exam test, so how are you going to do? Right, that'll be a very good question or very practical question to test whether you understand everything. Okay, I'll give you answer for this, and that'll be something you can think about it later. Okay, such as this protein T4 lysosine, and the T4 lysosine usually have to work in the lysogenic buffer or something at a high temperature. So you see, most of activity will be under 42 degrees. If you increase this enzyme's temperature to the higher temperature, it will cause this enzyme become denatured. Okay, so I want to add some disaccharide bond to increase their protein stability. So let's start to do all the randomized mutagenesis. Or they know that all those residue may require for their protein stability, especially this residue is for cysteine for the disaccharide bond formation. So you probably want to add more, increase more. So let's start to do all kinds of mutagenesis. Add one, add another two, but you will need to know how to assay its activity, such as in this case, they know that's a T4 lysosine, so they can provide substrate for this. And they know they want to assay thermal stability, thermal activity, so they can increase the temperature to test whether they still maintain its activity. Even this one is pretty good, right? They can maintain at a 48 degree, then still maintains activity. And later on, so when they add something more in this residue, they even can maintain 95% of activity. They can tolerate even higher temperature. So for this screening strategy, we'll give you a nice example. A lot of different engineering variants, you rely on good way for screening or selection, but you have to think about your screening strategy. You want to screen for higher activity or more thermal stability for your enzyme. And so after the random mutagenesis in the DNA label, you screen based on activity or thermal stability. It's up to you because I will not know what kind of protein you really want to have to adapt the new solvent or new temperature or new activity. Okay, so it will go back and forth, try and error. Maybe not just in this residue, maybe at the other position will be helpful for your purpose. Okay, so you do get all the protein. So if you want to increase the stability, just a xylanase. I pick up a xylanase to talk about it. So xylanase is the #1 protein for the biofuel production. Because all the pulp or paper industry, if they want to remove hemicellulose from plant, from wood, right, they need a xylanase. That's a rate limiting step to remove hemicellulose to make the wood pulp for biofuel production. But think about it, for the biofuel production, it definitely under certain quite terrible condition for the enzyme. Will be very high temperature, will be quite maybe organic solvent or acidic domain or basic buffer for this. So you think about it, then how to improve this enzyme to adapt this condition. So those are all important protein used for industry, like amylase, lactase, cellulase, glucose isomerase. They will have to work on hydrolysis of the starch for the brewing, milk processing, for sucrose, for plant or something. And all of them will be under quite difficult condition to use this enzyme. So you think about it, and you want to generate new form of the mutation for bleach boosting. So those enzymes, they need to accommodate at a 60 degree. They still have to maintain their activity because they need to do this kind of digestion under certain condition. Okay, so you will increase the disulfide bond at this condition. So for the native enzyme at a 60 degree, no one can survive. And then because of the disulfide bond, even you work at a room temperature, their activity even much better than the native protein. So they all rely on this kind of a knowledge we learned in the biochemistry. A lot of students told me that after the master training, that they want to go to the company for employee or for any engineer about that. If you really want to do something like this, I would suggest you to understand this part very well, because most company, they are making new version of protein, they are making new recombinant protein, they are making vaccine, modified vaccine through similar strategy. Okay, you look at all the example I'm going to show you, such as this enzyme, there will be something isomerase. You want the more efficient, half life is longer, then you will test to do all the mutagenesis from wild type asparagine and change to the surrounding and the isoleucine. They all rely on side-directed mutagenesis. So another example, I'm going to show you a lot of different examples. And all different examples, we have to go back to think about what kind of strategy they use. They use the error-prone PCR or use the side-directed mutagenesis. Most of them will use the side-directed mutagenesis because we already know which residue I want to add, such as the interferon. Interferon, E. coli express interferon. You feel that will be much cheaper, right? If we need the interferon for all the therapy, we don't want it to become very expensive and need to purify from cell culture, from pig, from any animal system. That'll be so expensive for the patient to use it. Recombinant protein will be very cheap. But unfortunately, you can see it in the slide. E. coli express the recombinant protein interferon alpha, only 10% of antiviral activity. So that's terrible. They almost cannot do anything. So later on, they figure out, okay, this kind of a dimerized or even oligomer, I mean, it will form oligomer between protein of the interferon alpha. So in E. coli, they will form dimer or oligomer. This kind of a protein will have a less activity for the human being. So after you know that, it becomes easy. You just remove the disulfide bond, right? Remove the disulfide bond. No matter you remove this one, remove that one, it won't cause any dimer or oligomer formation anymore, just purely by remove one residue. So those E. coli express interferon gamma become so powerful. Just remove one intramolecular disulfide bond, the recombinant form of protein interferon alpha become useful. Okay, another one like a serum protease. We know that the sublisting, that's Francis Arnold's first work about this detergent. The one is enzyme to fit into detergent company's purpose. But this is serum protease, sublisting. It has a one disadvantage. At that time, they spend a lot of time to solve it. Okay, they need the calcium for its activity. It needs a cofactor for its activity. If you think about this in history, that'll be so ironic. Anyone do your laundry, you still need to add calcium. If you would like to suggest your customer to add calcium in your laundry detergent, that'll be fun. That'll be a joke, right? So they understand this kind of a problem or bug immediately. So they need to remove this kind of requirement from the sublisting. So let's start to do all the mutagenesis, try out one mutation in this pocket. That's a 4 calcium binding. To change it to another residue. They still can maintain its activity, still maintain its stability, but then they don't need the cofactor binding anymore. So it will become good enzyme to put them into the detergent. They don't need any metal cofactor anymore. So, but that's actually a lot of work if you just rely on site-directed mutagenesis. Because you think about all the protein, that at least will have a 200 to 300 residue. So you can guess which one you want to do a site-directed mutagenesis. So most of the time, the first version, it was started from the error-prone PCR. And then find out good strategy for screening or selection for your activity, such as this one, will be no metal cofactor. The enzyme activity still work, right? So then test it, which one is the best one. And then later on, try to find out better version for that. Even you already know, okay, this pocket could be right. Then you start to do site-directed mutagenesis. So all this kind of molecular breeding will have a different level of the design. Okay, so that's another example for that. You can try to change alpha helical beta sheet structure from different one to test the enzyme's half-life, even without cofactor binding. Okay. Another interesting screening I'm going to introduce with you will be protein engineering in blood clotting quadralysis, this kind of a medical application. So usually when a patient has a stroke, sent into a hospital, ER, or outpatient center, they will inject some kind of a blood thinner to digest those fiber or fibrin accumulated in your bloodstream, especially in your brain, because this kind of accumulation will cause a problem. But this kind of plasmid, what is that? It's an active protease. This kind of blood thinner is an active protease. Theoretically, they will digest fibrin quickly. But later on, we'll see some kind of a problem here. The plasmid need to be activated from plasminogen. So after self-digestion, they will become active form of the plasmid. So plasmid will digest the fibrin quickly. So for the patient have the this kind of a fibrin aggregation, no matter heart attack or stroke, this kind of a plasmid, we call this streptokinase, is a kind of a clot dissolving agent. That will be for the myocardial infiltration, and they are very powerful for this kind of a emergency application. But later on, physician or start to feel some problem here. What is the problem? Okay, so plasmid or this kind of streptokinase, that's also protein. So they will digest their own protein immediately. So they will lose function for this kind of a protease immediately. So although plasmid can digest this kind of accumulated fibrin, but they also will lose their own function very soon. Okay, so decreased activity of the plasmid is a major bug for this kind of a strategy. So how to improve it? So the answer is quite simple. You just mutagenize streptokinase activity, still maintain is a protease, is a dissolving agent activity, but mutate plasmid recognition site is the protease recognition site, not big enough. So streptokinase still can do their work for the clot dissolving, but they will not be recognized by the plasmid. This kind of active form of the protease. So when you use this a new version of the streptokinase, you can cure patient, and then you don't have to worry about after you inject this protein, this streptokinase into the patient for the blood thinner, they will be useless very quick because in the old version, you see this kind of a problem after you inject. And the streptokinase activate plasmid, become plasminogen, become plasmid. That's also protease, right? And the plasmid will digest the fibrin. But the very quick plasmid will go back to digest streptokinase again. So it will become useless very quick, maybe in 5 minutes. So after 5 minutes, your drug become useless. But after you do the mutagenesis at the streptokinase, so let the plasminogen no longer recognize this guy. So they no longer will go back to digest streptokinase. So they will survive, and then the whole process will continue to work to sap patient to dissolve agent for any fibrin accumulation. Okay, so just modify plasmid recognition site on the streptokinase will be good enough. It also will not decrease the protease activity, but it will decrease plasmid's recognition to the streptokinase to improve its activity to become a long-lasting blood thinner. Okay, so the next example, I'm going to show you about sublisting. In the very beginning, Franz and Arnold, they use this enzyme, but they will need good or quick, simple strategy to screen which one is the best protease. So, sublisting is alkaline serine protease. Look at the alkaline serine protease. They do the shuffling, DNA shuffling, hope to get a good enzyme, new version of a sublisting quickly. But how are they going to screen? Because there's an alkaline serine protease. So milk is great. They are going to screen for protease activity. So they use 2% of skim milk. Milk contains a lot of different protein. But why we use 2% of skim milk? So no fat. So no fat at all. The kind of skim milk will basically coating on that paper, and it will spot your E. coli on the top. So the E. coli can digest the protein. So they will show clear zone here. So under specific temperature or time control and incubate with this E. coli, you will know which colony show higher serine protease activity. Okay, and you will pick up this colony for the further study or for the further mutagenesis. And later on, so especially currently, everyone loves this kind of high throughput strategy. So we probably don't use this kind of damn cheap screening strategy. We will use fluorescence dye or fluorescence substrate, and will give you different end product, and you can do different control. But all the concept, ideally, came from you need to know this enzyme activity, what kind of a feature you want to assay. So we will use a different kind of a chromogenic substrate, no matter with color, with a fluorescence, no matter you can monitor by your eyes or by computer or by video. All of them will work for your experiment. So let's talk about this part. So that's very important. Okay, let me ask again. You guys need a 10 minutes break or not? I can finish it pretty soon. Or you really want 10 minutes break after this slide? So you want me to finish as soon as possible? Finish as soon as possible. Okay, good. So I don't want to give you any break. If you need a break, go to the restroom. Go by yourself. You don't need my permission on that. So I'm going to finish all the slide as soon as possible. So you think about all the targeting treatment. We hope to generate antibody, any monoclonal antibody, to suppress, overexpress EGFR or TGFR receptor on the cancer cell to suppress cancer cell progression. Or we want to generate any antibody to recognize COVID-19, their code protein or surface antigen. So we will need the antibody. You see, this antibody rely on the variable domain, right? As a murine mouse antibody to get the antibody to recognize your specific region. But you know that most antibody, if from other species, you will have an immune problem because if you want to inject this antibody to a human being, they will recognize it as a foreign antigen. It's not my own antibody, so it will cause problem. Because what kind of problem you consider as a foreign antigen? So you will induce all the immune suppression, immune response to kill this antibody. It's not my own. So the best part, you want to have your own antigen to generate your own antibody with all native human protein. Which means you need to suffer from the antigen injection to get immune response, right? Sometimes it also could be very dangerous because this kind of a vaccination probably will cause a problem. And once you get a good antibody, can you share with everyone, such as after vaccination, I got a lot of antibody in my body. I want to share with my mom, my son, my husband. That's not possible, right? You cannot just sacrifice me to get the blood, to get all the antibody to cure other people. But you look at all the antibody structure, only variable region is important for recognition of the antigen. So is it possible we can generate chimeric one? Only this recognition side, but the other part still human part, right? Even now, we already know that which residue will be required or be sufficient to recognize antigen. We just do the mutagenesis, even recombinant protein, to express whole human antibody protein in E. coli with a specific mutation here for recognition of the COVID-19 surface antigen or for any specific receptor like a TGFR or EGFR. So you look at the oral targeting treatment here. That's a very important table to share with everyone, especially for students. If you are working on biomedical research, you definitely know that a lot of licensed therapeutic monoclonal antibody, and from chimeric IgG to recognize EGFR, until recently have a more and more so-called humanized IgG, also recognize EGFR something, or HER2 HER2 receptor, or TGF-beta or interleukin receptor. You see from murine IgG to chimeric IgG to humanized IgG. That's a huge advance for this field. See, from the chimeric to the humanized. Then that will actually be approved to reach the clinical application. Otherwise, even murine or any recombinant form of the IgG will be the big problem, will cause an immune problem in human being or in your patient. So you want to cure your patient, you don't want to kill them, right? But sometimes your drug, your antibody may risk to kill your patient. Okay, so the strategy for the direct evolution You also rely on the random mutation from PCR to generate a mutant library. And you will need to transform to the E. coli, then get a library, but you need a good strategy for screening or selection. Selection and screening, that's very different. If a screening, you want to check one by one for their activity. If a selection, you need to set up a certain criteria. Only some colony can grow under this kind of a stress, this kind of condition, like an antibody selection, or like an enzymatic selection, or thermal stability selection. And screening, you have to check one by one. Okay. So after that, even you will need a whole cell transformation for the analysis. Then pick up your true candidate. Okay. So all the novel protein design through this kind of strategy will be different. But a computer modeling is not follow traditional strategy or traditional concept to do mutagenesis. They give up all the traditional concept about the protein structure or everything. They purely generate or simulate protein structure in the computer program for the gene structure, for protein production, and eventually characterize their feature again, and to do this kind of unrelated protein selection for desired property, such as you want to have some protein or some new protein has a stronger recognition for certain protein substrate, then that's your desired property. So you don't have to decide it from the native protein. So you probably can combine several modules in the bioinformatics program to put everything together. It's just a Lego, this kind of a brick to put everything together. But you will need a good assay for this. That will work. Okay, later on, probably a few minutes, I'm going to show you some other fascinating example that are actually being used in the lab or in the hospital. Okay, you probably never think about a biosensor. Fluorescence protein came from this kind of strategy, site-directed mutagenesis. So I'm going to show you some structure function of a fluorescence protein and then those fluorescence proteins application. So that's definitely is my favorite picture from Roger Chan's lab. He used a little E. coli company to draw the Palm Beach for the San Francisco. Okay, you can see the red color and to see the blue color, green color. But all of them originated from the green fluorescence protein from the jellyfish. They are not so bright at all. That's not even applicable in the lab. So when he was invited to Taiwan to give a talk, he said that at that time, I just gave a proposal to the NIH and I got a job in the UC San Francisco. I pretty much worry every day I may be fired because I study green fluorescence protein. It's not really useful. It cannot cure any disease. But he feels this protein is so fascinating. It can label all the protein molecule to help us to visualize the organism's physiology in life. So that's his major purpose for his original research for the green fluorescence protein. And he showed that as a cover almost all spectrum of a different color for this protein. So because of his work, he's been awarded for Nobel Prize in 2008. Unfortunately, he passed away because of some kind of a heart attack problem. But everyone in this field will remember him forever. Okay, so his research started from a jellyfish, a green fluorescence protein. Individually, this green fluorescence protein is not very bright, very useful, even cannot be applied in the lab for the cell culture. Because if you think about jellyfish, they basically survive in the sea at around 18 to 25 degrees. So definitely will not be suitable for the higher temperature for the mammal in the cell culture for 37 degrees. So this green fluorescence protein will lose its fluorescence very soon. And then this green fluorescence protein, when they exposed to a blue light, like a UV spectrum, will excite to green fluorescence. But when you light up your cell culture with UV, you pretty much like doing a damage to them. So that will cause certain problem. So that's a lot of a problem for Roger Chen start his research. But the good news is that this protein is pretty small. You almost can tag this protein to all different part of the protein because it's around 25 to 28 kilodalton, 238 amino acids. Very easy to handle this protein. And its 3D structure is very clear. 11 strands of the beta barrel to form a three-dimensional structure, like a beer barrel. Okay. And then they also find out the whole green fluorescence protein actually only need a few residue. Glycine 67, tyrosine 66, and serine 65. And during this kind of excitation, UV exposed, you will make the cycling effect, dehydration, oxidation, to cyclize to make a maturation for the green fluorescence protein, chromophore. So for the whole process, it takes several minutes in jellyfish. So it becomes very useless in a lab. But for Roger Chen, she thinks that it definitely can improve. It's the first version of the protein because we already know the secret here. So we already know where's the chromophore. We know the whole reaction better. We know how to make this happen. So we know that you look at all the chemistry structure here. So how to make the cyclization, dehydration, and oxidation, you need to optimize pH, right? You cannot put your GFP protein in acidic condition. So even the buffer is not right, you will not see any bright green fluorescent protein in your experiment. That's a take-home message for this. Okay, and then they need the UV, this wavelength for excitation, and then it will emit around 509 nanometer green spectrum to see your protein. And the first version of a green fluorescent protein is not very bright, and the UV will damage your cell. UV equal to H mu, right? So the UV is around 300 something. So the energy is pretty high. So you will damage yourself. You want to label yourself, but you don't want to actually damage yourself. So they want to do mutagenesis to find out they don't need a 395 nanometer, or they can excite this chromophore formation. So they make the first version as 65T transition, new version of the GFP. This GFP protein, we even use it in the lab. We call enhanced version of the GFP protein to make it brighter, to make it more amenable in the lab. So it's the first version of the GFP protein has been formed. And other limitation will be what type of GFP protein will form very bad at a 37 degree because jellyfish basically they don't survive at a 37 degree. But if we want to use it in the lab, especially for the mammalian cell, it will not be good. So another round of mutagenesis will start. So okay, and the whole chromophore formation will take about 4 hours for the GFP protein to form correctly. Because for the jellyfish, they don't have to be super bright, right? Why they have to be super bright? They only need to make some very dim green fluorescence protein to help them to find out their way to go out for hunting. They don't have to make them become superstar in the sea, right? So they don't have to be very bright. So the 4 hours for the chromophore formation is quite reasonable. But for such as for our lab, we are working on cell migration. The total migration journey is only 6 hours. If it takes 4 hours for your GFP protein to 4 correctly to make a correct chromophore. The whole migration journey already finished. We cannot label anything. So that's another problem. So they need to engineer a lot of superfolded level 4 very quick, very soon to get this kind of protein. So you can see the whole kind of mutagenesis happen from the JP, emerald, superfolded, balsamic, green, wasabi. Wasabi is definitely a Japanese guy. He's a Japanese student to name. It's a new version. And so the GP protein from you can see the emission from the 353 to 488. That will not damage your cell, will be much better. And then their quantity or relative brightness compared with the original version will even become better. Like a superfolded GP will reach to 160 brighter compared with the EGP. So that's a very important improvement. They already summarized that in this paper. So you can see all the mutagenesis, they do it in this way, but only green fluorescence protein, people will not be happy. So they start to do more mutagenesis to give you a different spectrum. From YFP, we use it a lot in the lab, but YFP and GFP, they basically are from the same protein. The only a few residues are different, just like this. So the T203 will become YFP spectrum emitted. So if you want to use anti-GFP antibody to recognize YFP, it's okay, they can recognize. So if you want to distinguish them, to separate them, that's not possible. So the GFP antibody, you look at it with the YFP, the only a few residues are different. Antibody will misrecognize. They will think GFP and YFP are the same stuff. So they cannot distinguish GFP or YFP. Even BFP, blue fluorescence protein, or CFP, cyanide, they came from the GFP version. So that's common sense for that. So Roger definitely think that's not good enough. So they find out something from other species. Shanhu, okay, you will see that when you dive under the sea, it's a so pretty organism, right? And then they will show you bright red color. So at another spectrum for the fluorescence protein. So they will also have this kind of similar chromophore to form red fluorescence. Okay, so in addition to green fluorescence protein, we have another color now. So we'll have a ruby, apple, strawberry. Original will be red fluorescence protein. They even have a little bit different red, like a cherry. So even original one, the spectrum, their brightness is not very good. But until recently, for the ruby and the apple, the brightness is around 109, super bright. Okay, so in the lab, we still use strawberry and cherry or something. So if you want to compare with the green fluorescent protein, of course, it's not as bright as the green fluorescent protein, but at least it's another option to mark your protein or to mark your cell. Okay, so the protein engineering usually will involve 3 steps. You want to choose which protein you want to change, or you want to use a rational design, just like you already pick up a 3-catalytic triad, or you already pick up chromophore you want to change, or you want to do more randomized, like a sublisting. You don't know which one, which residue is a good or is activity. You will do randomized. The other one, you have to make a decision what kind of a mutagenesis method you want to use. Error prompt, DNA shuffling, anything you can think about. How you are going to evaluate these proteins variant with the improved property. You want to screen or you want to do selection. Just like this beautiful picture. It's not just a pure picture. They actually use the E. coli to draw this picture. So this E. coli, different E. coli, different version of the fluorescence protein give you different color. So such a simple strategy can be used for the screening, such as after mutagenesis, you want to make this GP protein become other spectrum, more yellow or more like a cell line culture. So you can streak all the E. coli to get a single colony. After you shine with the 488 nanometer spectrum of light, they will give you different color, except the green fluorescence protein. You will pick up this colony. So all rely on the strategy you want to use. So you think about it, all the different spectrum, different fluorescence protein. This chart is not for you to memorize that. This chart is for you to set up a different spectrum for excitation and emission. So when you like to tag some fluorescence protein, make sure you go back to check its excitation wavelength and the emission wavelength to get the correct setup for your confocal microscopy. Okay, so all of them, even you think they are very different color of the fluorescence protein, they only show a few residue differences. So they all rely on Z13 mutagenesis and randomized mutagenesis. Okay, so the summarize of the GFP protein, we all know that is so important for the modern biology because we probably can rely on in situ adaptation, rely on Western, rely on antibody standing in the fixed tissue. But if we want to know these proteins are dynamic, we want to do the lab imaging, you really have to take advantage of the fluorescence protein. Only fluorescence protein tag your component, tag your protein, you can see them in light. And especially this protein, the new version of fluorescence protein, they are still small, they are very stable, they can resist to a little bit high of the heat, alkaline pH detergent, or even they would be more tolerant about for the bleaching. And then their fluorescence property will absorb light from 395, that's the UV range, to increase it to 470 or to 480. They'll be more amenable for most of the tissue, won't cause any damage. And the excitation still maintained at the 508 nanometer. So that will be very important improvement for this kind of fluorescence protein. And they also will become quantifiable, and they will be reduced about the photofluorescence. So without excitation, they won't have autofluorescence for this kind of a protein. When you tag with your endogenous protein. So everyone love this kind of protein because once we tag with the GFP, we don't have to do anything. We can use the same antibody to detect all the protein. We even can see this protein to function, to act in like a chromosome. I can microtubule binding protein, mitochondria. I can watch this movie all the time. And this movie also tell us how exactly those reactions work, rather than we still have to have a lot of different speculation or different hypotheses to know how mitochondria work, how ER work, how microtubule work, how cell division or chromosome segregation work, right? We can add any drug, any inhibitor to see them in lab to see how those molecules to work together. So the final example I want to share with you guys about this fluorescence timer. Look at their title, why they consider as a fluorescence timer. Because this fluorescence protein will change color with time. That's so cool, right? They can generate red fluorescence protein later. In the beginning, they are green fluorescence protein. So this paper being published 20 years ago. So for this new application for this fluorescence timer, you look at this. So they basically use the error-prone PCR. See, error-prone PCR again. So to mutated to mutagenize the green red fluorescence protein. And this mutant form exhibit a very interesting phenomenon. They have a green intermediate fluorescent protein being screened and being visualized by the fluorescence microscope. So they will, mutation form will transmit from one color to another color with time. So because of this tool, they can change color over time. You can use this to label some protein to check their half life during development or during cell division. You will know how much time it takes for one process to proceed to another process. Okay, so this E5, only 2 residue replacement, is a new version of the protein, only from valine 105A and serine 197T. This kind of 2 mutation make such a dramatic effect. So you can see from green to red, it takes about near 48 hours. So if you can take a specific protein, you can observe their transition during cell division. And even you can guess how long this protein can survive for your biological experiment. Okay, I'm going to finish my lecture. That's a few take-home message that's important for your midterm exam. So you have to figure out what's the rational design. What does that mean for the randomized? So rational design of site-directed mutagenesis. What kind of strategy? What kind of example I give you in the PPT? Oh, I will upload my PPT later. And error prompt PCR is a major one for the randomized mutagenesis, right? And what's the error prompt PCR? What's the strategy for the error prompt PCR? What's the DNA shuffling? Why they can generate randomized library? And for your purpose, for your experiment, you want to choose a rational design or randomized design. And what kind of screening strategy or selection strategy you plan to use. So you need to show me your knowledge or your understanding for this lecture. So, okay, if you don't have any further question about this lecture, we are going to close today's WebEx lecture now. So if you have any question, leave in this WebEx lecture. If not, you are allowed to leave. Thank you. Any question? Okay, if no other question, you are free to leave. I'm also would like to close this session. I will post PDF file online. Okay, bye-bye.